Hello, this is Miss Dagenford, and I wanted to finish our conversation about the cell cycle. We had started on the diagram, and we have up here in our notes, we had our basic one, G1S, G2 mitosis. Now, my goal for this video is to help you understand why we would want to regulate it and how the cells regulate it, some of the new findings. And you'll notice the diagram that we have in our notes underneath this graph. This one doesn't have anything. I'm going to use one from the book that already has things labeled. If you'd like to hit pause right now, and you can go ahead and fill in your diagram, feel free to do so. And if you've done that, we can go ahead and just add some things to this. One thing you need to understand is that, mitosis needs to be regulated. I think we mentioned this in class, that when cells just divide willy-nilly, you get problems. You could get problems where we just have a buildup, like psoriasis, where the cells just pile up, or of course, the dreaded cancer, right? You can get a cancerous tumor or a benign tumor where the cells are just dividing uncontrollably. So cell regulation, mitotic regulation is important. And not to mention that not only because there are horrible things that happen when it's not regulated, but the fact that some cells should divide quite frequently. Your intestinal cells, your cheek cells, skin cells, all of these tissues divide quite quickly, as opposed to your nerves, where the pattern of how the nerves connect to each other are so important that you don't want those cells to divide. So, what can help the cells decide their fate? Are they going to decide or not? Well, first of all, let's go over some of the checkpoints. Now, there are some checkpoints that are designed to signal mitosis to go forward. Yes, go ahead and divide. But there are also checkpoints that are designed to put on the brakes. Okay, hold on, we gotta stop. And a lot of times, if that would be because there's been some damage. Either the cell has improperly sorted the DNA. They're not attached to spindle fibers. Maybe the cell didn't even copy the DNA correctly. Maybe there was DNA damage to begin with. Well, you don't want to copy damaged DNA. So how do you regulate this? Well, it has to do with these checkpoints. Now, the first one that I'm going to show you, and I'm going to go ahead and use green for go and red for stop. Not exactly very original there. So the first checkpoint, excuse me, the first checkpoint is at G1 as it goes into G2. So here's our little green light at G1. Now, this one actually is on the diagram. G1 is regulated, or at least we see this all throughout the cell cycle, but it starts in G1. It's going to start with the production of chemicals called cyclins. And so, what you're going to do in the cell, you have cyclins, and these cyclins they start increasing in number. Now, you might ask yourself, well, what's causing that? So the cyclins increase. Well, what is causing that? Well, these oftentimes are growth factors that are coming from outside the cell. So, for instance, if you damage your skin, maybe you get a little abrasion, you skin your knee. Well, those signals come from that damage. The damage is recognized, and then these growth factors signal, okay, those skin cells need to divide. So that's coming from outside that cell. And in fact, some of the first growth signals they found were from cells that are repairing damage. So these cyclins increase in amount. And so here you go, you can see the arrow, the cyclin accumulation. They start production here. And what they're going to do is eventually join with a molecule called CDK. So the CDK also increases in amount too. So this naturally happens. As long as you're getting growth factors, you're going to get these cyclins that increase the CDK. Now, the CDK stands for cyclin dependent kinases. And so these are kinases that are dependent upon cyclin. Now, what are kinases? Well, those are proteins that do something, they make it happen. And so they're signaling molecules. So you're going to see this almost in all of these go checkpoints, that the cyclins are being active, the CDKs. And so you'll notice they bind together. So as it increases in amount, you have this CDK and cyclin joining together, and it's at that point that we really push the cell to divide, the nucleus to divide for mitosis to happen. Now, that's all well and good, but what happens if there are problems? Well, let's go ahead and do a quick stop. So there's our little stop signal. Now, one, you're going to actually hear one that is going to be familiar. That's going to be later on. But in the G1 checkpoint, there is a very important molecule called P53. And P50, sorry, let me rewrite that. P53 has become quite the pivotal molecule here. We understand that this protein is going to make sure that the DNA is not been damaged. So P53 is a protein involved in checking DNA. So the important if then statement you need to know is if DNA is damaged, then P53 is going to stop cell division. Now, it could stop it completely, and if the DNA damage is completely irrevocable, it's just so damaged nothing can help, then the cell undergoes apoptosis, where it just completely dies. However, hopefully you repair. Hopefully you repair this. Repair the DNA, and then it can go forward. Now, these proteins, well, they're called tumor suppression. There's another one, retinoblastoma, RB, and it does a similar thing. It interacts with these to stop. Hopefully, you can repair, and if you repair, it goes forward. Now, because they discovered these in relation to tumors, and in fact, we call them tumor suppression genes. And you can, because they stop the cells from dividing if there is damage. Now you can keep going with this logical line of thought. If there's something wrong with P53, then that's going to cause problems with the body, because that allows these cells to start dividing. And as they divide, they have errors, and those errors cause rampant growth and problems. All right, well, let's keep going. We're going to talk about these a little bit in class, too. Well, let's do S. Now, in S, there's a go stop point, and the go stop points are these cyclins. So we have the cyclin CDK complex. And so, again, this increase is just going to continue that process. As they join together, that's going to accelerate or signal, okay, we got it, let's go. But an important thing happens during the S phase. Well, that's DNA replication. So an important checkpoint for S is to ensure that all DNA has been replicated correctly. So the signal here would be if there's DNA damage. So what we have are a couple of proteins, and they are again tumor suppression genes that encode these proteins. Now there's one called ATM, and it has a ridiculous name that we'll talk about in class. And what ATM does is it's a protein that then signals another one. It activates this protein, and this is where you might actually have heard this. The BRCA1 gene, and then that codes for a protein. And this is what you heard in the news where some women who know they have this gene mutation, if they have a mutation in that BRCA gene, then they might prophylactically have a mastectomy. They might remove their breast tissue because they know if you have a mutation in this BRCA1 gene, then you have a higher likelihood of breast cancer. So what now in a normal functioning BRCA gene, what this does is it promotes DNA repair. But you can imagine, just do your logic, if that's broken, then you're not going to be able to fix the DNA before the cell divides. So those are our two goes. Cyclin we see again, but then stop signals we're going to stop and try to repair that DNA. Now G2 G2 is going to be after DNA replication, of course. So we're still going to need to check for DNA damage. So we have our go signals, which is again our cyclin CDK complexes. But now we have another stop signal, and the stop signal is one that we've seen before, and that's P53. And P53 is again going to be checking on DNA damage. It's going to be checking on our organelles, whether or not we have plenty of them and able to divide in resources. Now there is one more checkpoint, which you think at this point in time oh, okay, well everything should be okay, but we've got to worry about one more thing, and that is during mitosis itself. So during mitosis itself, we have again the CDKs, and the CDK and the CDK complex together. That's called the mitosis promoting factor. So it just keeps increasing and increasing and increasing. You can see this arrow getting bigger, and at mitosis, well, this is causing okay, we gotta start mitotic division. So we have our CDK cyclin complex. And again, together that is called the mitosis promoting factor, sometimes called maturation promoting factor. Well, but so what's the stop? Well, the stop is a protein called MAD, and what the protein can do, if it's working properly, what it does is it checks that all of the chromosomes are attached to the spindle fibers. And so if everything is okay, then you go from metaphase to anaphase, the chromosomes separate fine. However, if one of the chromosomes haven't if they haven't attached to the spindle fibers, well, when it comes time to try to split, if they're not split, if they've not been attached correctly, then you might have a situation where one chromosome, completely duplicated chromosome, gets dragged to one side and it doesn't split. So this checkpoint makes sure that the chromosomes are attached to the spindle fibers. And what MAD does, it activates a protein that breaks down the what attaches the two sister chromatids together. So proteins between the sister chromatids break down, and eventually you get to the point where the sister chromatids can be pulled apart, but that only works if both sister chromatids are attached. So that's the last checkpoint. Well, hopefully you can see the connection between some of these molecules and the regulation of the cell cycle. We're going to talk about how this impacts cancer and the growth of those cancerous cells. Have a lovely day.